Thank you so much for coming. My name is Julia Mizo, and I have been studying the effects of changing ocean salinities on the development of the tropical sea urchin Echinometra mathii. So I chose to study sea urchins because I was interested in learning more about our marine life. Growing up in Hawaii, I feel a special connection to its beaches and the ocean life within. And through my research project, I was able to study something I was both passionate and concerned about. Considered to be a keystone species, sea urchins play a key role in the maintenance of their ecosystems. They help to keep an ecological balance, and a lack of sea urchins within an environment can be detrimental to the health of coral reefs. As grazers, sea urchins consume algae to regulate overgrowth on coral reefs. A loss of sea urchins could equate to the uncontrolled spread of algae that, in turn, inhibits coral reefs from using the sun to photosynthesize. As a result, the habitat that coral reefs provide for other species could cease to exist, causing a domino effect of susceptible marine life living in the ocean. Echinometra mathii are one of the 8 sea urchin species found in Hawaii's coral reefs, which is home to over 1,000 species of marine life, and around 300 of those are endemic to Hawaii. Not only are coral reefs important for maintaining biodiversity in our oceans, but they also protect against coastal erosion. Without them, Hawaii would be vulnerable to coastal hazards and ecological dangers. One alarming result of climate change is the effect that it has to the Earth's water cycle. This contributes to changes in the salinity levels of oceans at the surface level. It is expected that the global water cycle will intensify in the future, leading to changes in rainfall patterns. Hawaii is estimated to experience more frequent and intense rainfall, which will dilute the seawater and decrease salinity levels. Influxes in evaporation at the ocean surface will increase the salinity. And along with that, as the temperature of the ocean increases, so does the salinity, especially at shallow surface regions. Human activity can also affect salinity levels. This includes land use, coastal development, and pollution, which could lead to nutrients and runoff to enter the ocean and alter the salinity. Along with that, sea urchins are especially vulnerable at the embryonic stage. Their predominantly calcium carbonate structure at this point in their life causes their development to be heavily influenced by changes in their environment. Embryonic reactions to these changes serve as good indicators of how it will affect the adult population and their species as a whole. Thus, my research focused on the development or lack thereof of Echinometra mathii embryos, and my research question becomes, how does changing the ocean salinities affect the embryonic development of the tropical sea urchin Echinometra mathii? The results that I gathered from my study provided insights on the boundaries of acceptable or suitable salinity levels for proper sea urchin embryo development. In order to assess this, I had to determine how different salinities compared to a salinity match treatment, which was the control condition. My goals for this study was to culture embryos in 4 salinities, observe their development at 24 and 48 hours post-fertilization, and analyze their development. This is a timeline of the first 2 days of a sea urchin's life, starting from the cell stage, and these are all pictures that I took through a compound microscope. I was able to observe the mitotic 2 and 4 cell stages, then the morula and blastula stages, all within the first 24 hours post-fertilization. I classified having reached the blastula stage by the formation of purple pigment and mesenchyme cells near the bottom of the embryo. From 24 to 48 hours post-fertilization, I observed the gastrula stage, defined by the development of an archanteron, the prism stage, defined by the development of spicules, and the pluteus stage, which was defined by the development of arms. I hypothesized that embryos in the highest salinity would have the slowest development at both 24 and 48 hours post-fertilization when compared to the control condition. There were 4 main steps to my study. I first collected sea urchins, then fertilized them. I then created 4 salinities to represent different ocean conditions, and lastly, I observed and analyzed their development in each of the salinities. Sea urchins were collected in the shallow reefs at Ala Moana rock piles of around 1.5 meter depth. They were collected with a handheld scraper to ensure no damage done to the coral reefs. They were put into a plastic bag and allowed 15 minutes to acclimate before being fully immersed into the tanks in our lab, and they were fed a constant supply of algae that was collected from the same sample site. Sea urchins were injected with 0.5 molar potassium chloride solution near the mouth cavity to induce spawning. This was a difficult process because we could not determine the sex of the urchin until after they had been injected. Female urchins released orange eggs, while the male urchins released white sperm. The female urchins were inverted over a cup containing filtered seawater, and the eggs were collected at the bottom. Sperm was pipetted off the male using a micropipette. The sperm was diluted, then added to the egg mixture. The eggs were checked under a microscope for good quality and egg density. Fertilized eggs were then decanted and made into a 30 milliliter embryo slurry. The embryos were observed in triplicates of 4 salinities, 32, 35, 38, and 40 parts per thousand. The 35 parts per thousand served as the control condition closest to the salinity of regular ocean water. 32 was the low, 38 was the middle, and 40 was the high salinity. Each of the salinities were made by adding different amounts of an artificial ocean mix to 30 milliliters of filtered seawater. Salinities were measured using a handheld refractometer. Triplicates of each salinity were contained in a 12-well plate that looked like this diagram, with each well containing 5 milliliters of the desired salinity. 150 microliter aliquots of the embryo slurry were pipetted into each well, and the embryos were kept in an incubator at 25 degrees Celsius. A compound microscope was used to observe the embryos. 150 microliters of each well were pipetted onto a depression slide and examined. To ensure randomness and a lack of bias, the first 10 embryos seen were classified and recorded. Since there were triplicates of each salinity, there was a total of 30 embryos that were counted from each salinity. The counting was done at 24 and 48 hours post-fertilization. This is what a normal development pattern would look like, which was predicted and followed by the control condition. The data collected from each salinity allowed us to make comparisons to how their development differs from the control. My count data is displayed in a bar graph with the salinity on the x-axis and the number of embryos counted on the y. The orange represents the number of underdeveloped embryos, green is morula, and blue is blastula. At 24 hours post-fertilization, the blastula stage is the predicted and normal development stage. The control salinity is the bar furthest to the left. As shown by the graph, it has about the same number of embryos that had developed to the blastula stage as those in the medium and high salinity. However, when the control is compared to the low salinity, we found that there is a statistically significant difference in development to the blastula stage. The statistical analysis was done using a Fisher's exact test, which gave a p-value of 0.017, which is less than 0.05, so we could determine the difference as significant. At 48 hours post-fertilization, optimum development was to the pluteus stage, which in this graph is represented by yellow. Again, the control condition is the one furthest to the left. When compared to the medium salinity, they both have a majority of embryos that had developed to the pluteus stage, and their differences were statistically insignificant. However, when the control is compared to the low and to the high salinities, there is a statistically significant difference in the number of embryos that had reached the pluteus stage. We again use a Fisher's exact test that gave a p-value of less than 0.001, confirming its significance. Along with that, we can see that there is a difference between the low and the high salinities. While around a third of the high salinity embryos had reached the pluteus stage, none of those in the low salinity had done the same. Most of the embryos in the low salinity at this time were either dead or still in the blastula stage. Meanwhile, the high salinity contained embryos in the prism stage, which precedes the pluteus and could indicate that they are still developing. The low and high salinities impacted embryonic development at different life stages and in different ways. At 24 hours post-fertilization, we saw that it was only the low salinity that had embryos with a slower development. Then at 48 hours post-fertilization, embryos in both the low and the high salinity had slow development, but it seemed that the low salinity had either stopped completely or were at an even slower rate. Data from the experiment suggests that salinity affects development in the early and late stages. Any delay in the development process to the Plutia stage will mean they will find difficulty in attaining food, escaping predators, and gaining the ability to move. Problems such as these during this stage could result in a decreased chance of survival of the sea urchin. As a result, bottlenecks may occur to their population. And if this were to occur in a specific ecosystem, it could lead to critical issues such as a complete end to their population. It is unclear by exactly what magnitude the salinity of Hawaii's oceans will change by in the future, but it is probable that we will see changes. Sea urchins are just one part of our coral reefs, but they serve an important role in our ocean's ecosystem. Due to certain limitations in our lab equipment, the quality of our results were limited. These factors included the zoom level of the compound microscope and the consistency of temperature in our incubator. Future studies could increase the range of salinities in which the embryos are reared. From this data, a performance curve of salinities on embryonic development could be created. A long-term study would also be required to better assess whether sea urchins could adapt to these changes. Future studies could also test multiple stressors either individually or simultaneously. Climate change affects more than ocean salinity. Near future conditions could see dramatic changes in temperature and acidity as well. I would also like to give some advice to future students, which includes being accepting of feedback and open to criticism, staying organized, and choosing a topic that you genuinely enjoy. This is a year-long class and project, so it's important that you find something that makes you excited to go to class and lab every day. Lastly, I wanted to thank a lot of the people that have helped me this year. Thank you to Dr. Chan and Mrs. Kobayashi for guiding me through my first research project. There are not many teachers who are willing to spend Christmas and New Year's with me in the lab, so thank you so much for all of your time and help. I also want to thank Dr. Amy Moran for her willingness to be my mentor and for all of her knowledge and wisdom on sea urchins. Thank you also to Mr. Hill for helping me with the statistical analysis in my project, and my family for helping me a lot in the beginning of my project, taking me to the beach and making me aware of the environmental challenges that sea urchins face. Lastly, I want to acknowledge my honors independent research classmates who are all very smart and inspire me to work hard and have fun with my project. These are my sources and my literature. Thank you.